Are you tired of waiting months, even years, for your compost to be ready? What if I told you there's a revolutionary Japanese technique that creates nutrient-rich, living soil in a fraction of the time? This isn't just about faster growth, it's about transforming your garden with accelerated nutrient cycling and incredible microbial activity, making your plants thrive like never before. Say goodbye to slow composting and hello to a garden that practically grows itself. The composting conundrum: why does speed really matter? Traditional composting serves gardeners well, building humus-rich amendments that improve soil structure and fertility over time. The challenge honestly lies in the timeline. Hot composting requires 6 to 8 weeks of active management with regular turning, temperature monitoring, and proper carbon to nitrogen ratios. Cold composting extends this timeline to 6 months or even a year before materials break down into usable amendments. For gardeners working with tight growing seasons or rapid succession planting, these timelines create bottlenecks that, limit productivity. So, the biological reality of traditional composting involves thermophilic organisms that actually dominate during the hot phase, reaching temperatures of about 130 to 160 degrees Fahrenheit. While these high temperatures are great for killing off pathogens and weed seeds, they also, unfortunately, eliminate many beneficial microorganisms that plants really depend on for nutrient uptake. The finished compost, well, it requires a curing period where mesophilic bacteria and fungi recolonize the material. This step adds weeks or even months to the whole process. Urban gardeners, especially those with limited space, face some extra challenges here. Traditional compost bins need a significant amount of square footage, and, honestly, they can generate odors that neighbors might find pretty objectionable. So, modern gardening, it really demands faster solutions. Intensive bed systems and succession planting, they require constant nutrient inputs to keep up productivity. And, container gardening can actually exhaust soil media within just one season. Small-scale market growers, well, they need reliable and rapid soil building to keep production moving, and that's without relying on expensive commercial amendments. The gap between traditional composting timelines and contemporary gardening needs, it creates an opening for alternative approaches, ones that can accelerate the process while still maintaining or even improving biological quality. Now let's talk about aeration fermentation, which is the Japanese approach. This technique actually draws from bokashi composting principles that were developed in Japan during the 1980s by Dr. Teruo Higa at the University of the Ryukyus in Okinawa. So, rather than relying on thermophilic decomposition, this method actually uses controlled fermentation with beneficial microorganisms to pre-process organic materials. The key difference lies in the process. Traditional composting oxidizes organic matter through aerobic decomposition, while fermentation uses anaerobic processes that preserve nutrients rather than releasing them as gases. The fermentation process employs effective microorganisms, which is basically a consortium of beneficial bacteria, yeasts, and fungi that work together to break down organic materials. Lactic acid bacteria dominate this mixture, creating an acidic environment that suppresses putrefaction and pathogenic organisms. So, yeasts contribute enzymes that break down complex carbohydrates and proteins, and, photosynthetic bacteria enhance the microbial community by producing beneficial compounds that support plant growth. Research from Japan's National Institute for Agro-Environmental Sciences demonstrated that fermented organic matter increases soil microbial biomass by 40 to 60% compared to conventional compost. The fermentation process preserves nutrients that would otherwise volatilize during hot composting. Nitrogen retention improves by 25 to 35% because the anaerobic environment prevents nitrogen conversion to ammonia gas. Phosphorus and potassium remain in more plant-available forms, bound in microbial biomass that releases slowly as organisms die and decompose in the soil. Setting up your fermentation system starts with, well, the foundation, which requires an airtight container that excludes oxygen during the initial fermentation phase. Food-grade 5-gallon buckets with tight-fitting lids work excellent for household applications. For larger operations, you can use 30-gallon containers or specialized bokashi bins with drainage spigots. The key specification is an oxygen-free environment that allows lactic acid bacteria to dominate over aerobic decomposers that would create odors and nutrient loss. Material preparation starts with chopping or shredding organic matter into pieces smaller than 2 inches. Smaller particle size increases surface area for microbial colonization and, accelerates fermentation. Kitchen scraps work perfect, including items traditional composting excludes like meat, dairy, and oils. Garden waste, spent crops, and even paper products can be fermented. The diverse input creates finished material with broad-spectrum nutrients and microbial diversity. The inoculant consists of effective microorganisms mixed with a carrier material. Traditional recipes use wheat bran or rice bran inoculated with EM solution, creating a dry mixture that spreads over organic materials. For a 5-gallon bucket system, you'll want to use about 1 cup of inoculated bran for every 2 to 3 inches of organic material. Layer the materials in the bucket, adding bran between each layer, and make sure to press everything down firmly to exclude any air pockets. The compression step is really critical since trapped oxygen pockets can let aerobic organisms compete with your fermentation bacteria, leading to foul odors and, honestly, reduced quality. Moisture content is something to pay attention to, but it doesn't need to be as precise as with traditional composting. The materials should feel damp, but not, dripping wet when squeezed. Excess moisture will drain down to the bottom of the container, creating a nutrient-rich liquid. This leachate can be diluted at a ratio of 100:1 with water and used as a soil drench or even as a foliar feed. It contains dissolved nutrients and beneficial microorganisms, so it provides immediate plant nutrition while the solid material keeps on fermenting. The sealed container sits at ambient temperature for, about 2 to 4 weeks. Warmer temperatures can speed up the process, with ideal fermentation happening somewhere between 68 and 77 degrees Fahrenheit. The container can be kept in a garage basement or even under a kitchen sink. During fermentation, the pH drops to around 3.5 to 4.0 as lactic acid builds up. This acidic environment helps preserve the materials in a pickled state, instead of letting them, rot. A sweet-sour smell, kind of like pickles or silage, means fermentation is going well. On the other hand, if you notice foul, rotten odors, that usually signals failed fermentation, maybe from not enough compression, not enough inoculant, or some oxygen sneaking in. After fermentation is finished, the material needs a transition phase before it's actually available to plants. The acidic, pickled organic matter has to interact with soil organisms, which will finish the decomposition process. To get started, you'll want to bury the fermented material in your garden soil at least 2 weeks before planting, making sure to mix it into the top 6 to 8 inches. Over the next 7 to 14 days, there's a transition from anaerobic fermentation to aerobic decomposition, as soil microorganisms move in and help neutralize the acidity. If you need to use the material right away, just mix the fermented material with finished compost or aged leaf mold, using a ratio of 1 part fermented material to 3 parts mature compost. This buffering step, lets the acidic fermented matter blend in without shocking the plant roots, while also providing the diverse microbial community needed for the final decomposition. The result is a growing medium that's rich in both stable humus from the compost and labile nutrients from the fermented materials. Container gardeners can, create potting mixes using about 20 to 30% fermented and transition material mixed with quality potting soil or coconut coir. Field applications involve broadcasting fermented material over beds and then incorporating it with shallow tillage or cultivation. Application rates of 10 to 20 pounds per 100 square feet provide significant fertility without causing excessive acidity. For established perennial beds where tillage just isn't possible, simply spread fermented material as a top dressing and cover with, say, 3 to 4 inches of regular compost or mulch. The results and beyond. Gardens using fermented amendments show measurable differences in plant performance. Research from South Korea's Rural Development Administration compared vegetables grown with fermented organic matter against conventional compost controls. Plants receiving fermented amendments showed 30% faster early growth rates, which is attributed to higher populations of beneficial soil bacteria and, readily available nutrients. Root systems developed 25% greater mass and penetrated 15% deeper, accessing water and nutrients from larger soil volumes. Nutrient analysis reveals why fermented materials outperform traditional compost. Available nitrogen in fermented amendments tests 40 to 60% higher than finished compost because the fermentation process preserves nitrogen in microbial biomass rather than releasing it as ammonia. So, phosphorus solubility actually increases thanks to organic acids produced during fermentation, which chelate phosphorus and make it much more available to plants. Plus, the microbial biomass itself acts as a kind of slow-release fertilizer, breaking down gradually, over several weeks to provide steady nutrition. This technique is really flexible, scaling easily from apartment balconies all the way up to small farms. Urban gardeners really appreciate the odor-free process, which fits conveniently under kitchen sinks and, turns food waste into fertility, without attracting pests. Small-scale farmers, are using larger fermentation systems to process crop residues and create fertility inputs without needing all the land area that traditional compost windrows require. The processing time of just 2 to 4 weeks, it really allows for rapid cycling of materials. That means you can produce finished amendments multiple times each season from the very same container system. Subscribe to the Living Soil Lab to keep exploring these innovative techniques that, honestly, accelerate soil building and intensify production, all without synthetic inputs. Go ahead and share this guide with gardeners who are ready to transform waste into fertility, way faster than traditional methods allow. Together, we're showing that small-space, rapid-cycle systems can produce the living soil that feeds abundant gardens through every season.